Okay, good day, everyone. Thank you for being with us in this final presentation of the CEiRSI Pilot 4. This pilot is on the transcatheter aortic valve implantation, where we studied the frame release and pre-post implantation blood flow models predicting aortic stresses and valve leakage for In-Silico trials. But before we start with the presentation itself, I will give the floor to Alex Fangri from Manchester University, who will give us some words about the project and the Airlock project.
Yeah, thank you very much, Marta. And I'd like to welcome everyone to the UK CEiRSI, or Centre of Excellence in In-Silico Regulatory Science and Innovation training webinar series that are proudly supported by Innovate UK, the Medical Research Council, or MRC, and the Medicines and Healthcare Products Regulatory Agency, or MHRA. As the UK's first centre of its kind, we pioneered the use of In-Silico methods to enhance regulatory science, innovation, and our webinars will delve into the latest advancements, best practices, and applications of In-Silico approaches. We hope to foster collaboration and knowledge sharing among stakeholders, as you will see today, so that we can drive progress in healthcare and medical research. And more broadly, we will put some links as well on the webinar for you to join our network through various channels that we have. And we hope that you can contribute as well and get inspired by this webinar. And thanks to Marta and the rest of the team for these pilots. And I'll hand over the floor to you, Marta.
Thank you, Alex. And I will ask Gidan to share the screens, the screen with the presentation, so we can start. Yeah, thank you, Gidan. Well, we're gonna start with the next slide, please, on the agenda. Today we will talk about show the members of this pilot who intervene in all the discussions and in the work. We will give an overview on the schedule of the pilot. We will talk about what is a TAVI and to explain a little bit the general concept of a TAVI device and the therapy and the general procedure. Also, we will talk about the structure and the methodology that we worked. We will address why this pilot is important for regulatory perspective. Also, we will talk about the description of the question of interest and context of use, the bench testing, clinical aspects, and some other technical aspects. And of course, we will discuss on risk assessment and In-Silico credibility evidence. And also why it's important of the predicate clinical trials, and you will see why it's so important. And also the interaction of impact of artificial intelligence in computational modeling and simulation, and giving an overview of discussions, difficulties encountered, important points, and the final remarks, and then we will proceed with the questions and answers. The next, please. And here we have the panelists. With me leading the work is Dr. Ali Sarami, the lecturer in cardiovascular biomechanics and on the Manchester University. Dr. Yidan Xue, who is research associate in the university. And we have also Dr. Adesola Ademiloye, who is program director and senior lecturer in Swansea University. Dr. Renel Bombin, associate professor for cardiac surgery and clinical director from UK CEiRSI. We have Mark Gambridge, head of clinical investigations on MHRA, Alexander McLaren as a benefit risk assessor in MHRA, and Dr. Cahill McVeigh, that is research director, global technology and innovation in Medtronic, and Dr. Pawan Kumar Pandli, and is research associate in University of Manchester. We go next slide, please. And here you can see all the parties intervened in this pilot project, and the best and the biggest sponsors, of course, is Innovate UK and Medical Research Council, and we had the collaboration of Exponent, MHRA, Medtronic, Edwards, Manchester University, of course, QTech, and the University of Edinburgh. Next, please. We need to do a disclaimer. So you will see a lot of references of specific manufacturers, devices, or products, but these are only for this presentation to provide solely for the illustrative purposes. They don't imply any endorsement of recommendation of preference for our organizations. And any example that you see are intended to support discussion and understanding of regulatory and the technical concepts exposed here, and apply to the use of acceptance of in-silico methods. Next, please. Now the pilot members, I can say first that the CEiRSI Pilot 4 initiative was organized through a structured series of biweekly meetings and dedicated workshops that ensure the consistent progress of methodology development, paper drafting, and final presentation preparations. The coordination was facilitated via shared repositories, Slack channels, and scheduled rehearsals with clean timelines and deliverables, such as the draft of a paper and webinar deck. The pilot brought together a diverse group of stakeholders reflecting a balanced presentation across the industry, regulatory bodies, competent authorities, and academia. And the industry participants included device manufacturers and technical consultants. Regulatory experts were drawn from agencies such as MHRA. And also academic contributors represented leading institutions in computational modeling and medicine. The multidisciplinary engagement ensured the comprehensive coverage of technical, regulatory, and scientific perspectives, strengthening the credibility and impact of the pilot output. The next, please. And here you see the list of members that collaborated in all a series of the discussions, and it's not necessary to mention name by name, but you can see later on when you receive copies of the presentation. We go further. Next, please. And now we will see an overview of the schedule of the pilot. And next. So here we started in the pilot in April 14. We made an introduction structure of the bibliography that we consulted for that. And then immediately after, in June, we started with the drafting a paper that will support all the discussions and findings that, of these pilots. And then we have a revision in September and through October and November. And we want to present the draft paper as soon as possible. Our intention is to publish something in December before the season holidays. Next time here is how we are today. We along our discussions, we presented as well and scheduled in order to prepare this final presentation. And as you see, the paper will be public very soon. Next, please. And now I will give the floor to Keihal Magbale, Research Director and Global Technology and Innovation from Medtronic, who will give us an overview of the TAVI device and the therapy and general procedure. Thank you.
Thanks, Martha. I think with the introduction done, it's good just to skip to the next slide and we can get going. So I'll be giving an overview of the anatomy, the disease state, and the therapy that is, as Martha described, the focus of Airlock 4. we'll start with the basic anatomy that you see here. So the heart has 4 valves. One of those is the aortic valve, which we'll be talking about a lot here. That opens to allow oxygenated blood to leave your left ventricle. That blood travels to the rest of your body. Of course, the same valve closes to prevent backflow, and it does that opening and closing once every heartbeat. Some people, as they age, suffer from aortic stenosis. There's a little video of that on the top right that you just saw play, where the valve, it really becomes calcified and stiffens. That results in a resistance to blood flow through that valve out to the rest of the body, and that it manifests itself as a high transvalvular pressure gradient. The focus of our presentation is on TAVR, which is, or sometimes called TAVI, so transcatheter aortic valve replacement or transcatheter aortic valve implant, which is, it's essentially an artificial valve which is implanted inside that diseased aortic valve and replaces the function of that diseased valve. Little schematic in the bottom right really just shows you that we can avoid open heart surgery because, as the name would suggest, with TAVR, it's delivered via a catheter. The valve is crimped or compressed inside that catheter. The catheter delivers it or transports it to the implantation site where it's deployed. And we'll talk a little bit more in detail about how that happens. But if you go to the next slide, please, Eden. So our Airlock discussions were really focused on this device, and there is an ISO standard which really helps manufacturers. It guides them on how to test the device on the bench, and it provides guidance on things like animal studies and clinical studies. And what you should know is that that standard also allows us to justify omission or abbreviation of clinical investigation in circumstances where a minor design modification is being made. So that was kind of the starting point of our discussions. We were asking ourselves this question. So in this scenario of a modest design change to the device, which the other presenters will describe in more detail, can in-silico evidence be used to form part of this justification? So that's why we're here today. But yeah, before we get into a deeper discussion on that, we'll cover a little bit more about the disease and what the treatment options look like. So this is what we're dealing with. It's like I said, it's a disease of the valve. The valve, which it really becomes more prevalent with age, and there's some figures on the right-hand side there. That disease, it makes the heart work harder. It makes it harder for the heart to pump blood. The heart being a muscle, if there's more resistance to that, it grows to compensate. That results in a thicker ventricular wall, smaller capacity in the ventricle, and ultimately it can lead to heart failure. Next slide, please. First line of defense is meds. A catheter balloon might also be capable of stretching out that diseased valve to restore some of the functionality. Third option, surgical valves. They've been around for a long time at this point, around for decades. They're very effective if you're strong enough to survive the very invasive procedure, which you have a picture of coming up. Transcatheter valves in the bottom right, that's what we're here to talk about. They're minimally invasive solutions, obviously the focus of this talk. So let's go to the next slide. This is what a surgical replacement looks like. It's obviously it's a very serious procedure, an open heart procedure. It's something we want to avoid, especially for older, frailer patients. Patients on cardiopulmonary bypass, there's a machine breathing and pumping their blood for them. So think in terms of, over a week in hospital, 8 weeks for the wounds to heal, 6 months before regular activity could be resumed. Next slide, we'll talk about the alternative, which is TAVR. Those patients are typically back up on their feet and back to their usual routine in a couple of weeks, 1 to 2 weeks. There's lots of different types of TAVR devices on the market. They generally share a kind of common architecture that you see on the right-hand side of something that looks like a stent that we refer to as a frame, a metallic structure. That's there to support the soft valve. It houses that valve. That valve consists of flaps of tissue, essentially, that are harvested from pigs or cows, and specifically from the pericardium sac that surrounds those animals' hearts. Next slide, please. There's a couple of videos here in the bottom, if maybe you can just click them to play them. So the catheter that delivers a device, it can be introduced at different locations on the body, percutaneously, obviously. And there are really two different types of TAVR that you'll come across. Self-expanding devices, bottom left, which it makes use of a super elastic nitinol frame to expand the device and to anchor it into the anatomy. Bottom right, you see a balloon expandable device, which also anchors in the anatomy, but now through an elastic plastic frame deformation, which is really driven by balloon inflation. Next slide, please. So in terms of indication, these devices are typically indicated to treat severe aortic stenosis, even in bicuspid aortic valves, and also in failed surgical and failed TAVR devices, which you'll sometimes hear referred to as redo or TAV and TAV. It's a very fast-growing market, over 200,000 patients treated per year. If you go to the next slide, it gets into more details on the breakdown. So over half of those are in the US, but a growing number, we've probably tipped over the 10,000 mark per year in the UK and over 30,000 per year in Europe at this point. That is a little old. Next slide, please. The devices are getting better with each generation. Performance is improving. You'll see additional design features being added into the devices from most manufacturers. And I mean, what this slide's intended to convey is that manufacturers often favor an approach where iterative modifications are made to these clinically proven platforms to progressively improve aspects of the design based on, the real world experience, learnings, and feedback to the field. So the devices first received CE mark around 2007, and FDA approvals really followed a few years later. At this stage, thousands of patients have been enrolled in clinical trials, and we're really continuing to learn about these devices. So next slide, please. So what we know is that, complications remain. The primary issues that patients might face are things like stroke or conduction disturbance, which can occur when the implanted device interacts with the cardiac conduction system. There's a nice diagram on the left that shows where these devices are deployed relative to the membranous septum and the left bundle branch. So these are areas, best avoided in terms of conduction disturbance. The other one, paravalvular leakage, that really means leakage between the device and the anatomy, usually during diastole when the valve is closed. Annular rupture due to overexertion of load on the anatomy during delivery, and coronary obstruction if the device itself blocks flow into the coronary arteries, which is obviously important to feed the heart muscle itself, or other vascular complications. So that's kind of a crash course on TAVR, and at this point we should know enough about TAVR for Yidan to be able to describe the study methodology. So I'll hand over to Yidan.
Thanks, Cathal. I'm Yidan. I'm a research associate from the University of Manchester, and I will give you a brief introduction on the structure of methodology for this pilot scenario. Yeah, so here we have a hypothetical scenario where we are a company with a successful TAVI device with a history of competitive clinical success on the market. And right now we have a strategic aim to further reduce the rate of pacemaker implantation, which is typically caused by the conduction disturbance due to contact pressure from TAVI device frame to the aortic root, so that the device can be more marketable. And so an evolution of the existing design with a small change to the lower section of the device frame has already successfully passed the preclinical assessment. And the company is now engaged in clinical development to plan for the future trial that will be used to gain market access for the redesigned device. And during this process, the company has used in-silico models and tools to do the preclinical assessment. And so they are wondering whether in-silico evidence can be used to reduce the clinical trial size and therefore accelerate the time to market for the new design. So the overarching question of interest for this pilot is, can a minor design change to the lower section of a TAVI design frame, specifically near the aortic annulus, can be approved with a reduced patient recruitment size by leveraging in-silico evidence? And to answer this question, we break down today's presentation into 5 parts. So firstly, we will discuss the regulatory and business interests of this pilot. So we will discuss what clinical data is required, which points need to be considered, and what's the general MHRA review of a clinical investigation for a TAVI device, and what are the ethical considerations. Then we will analyze all the TAVI risks and found the risks that can support that can be supported effectively by in-silico evidence. So we will discuss how to define the context of use for this in-silico regulatory submission, how these context of uses connect to each other, and how to consider existing bench testing and clinical trial evidence. And then following this context of use, we will do the model risk assessment consisting of model influence and the decision consequence for the deployment, conduction disturbance, and the parallel leakage. And with this model risk, we will recommend credibility evidence that needs to be collected to support the use of these in-silico models in the regulatory process. And we will discuss how to use predicate clinical trial and best bench testing results, like to reduce the model influence, and how to consider the model and parameter uncertainty. And finally, we will look ahead to discuss the artificial intelligence and their impact on computational modeling and simulation. We will focus on the current applications and the barriers of AI models in TAVI in-silico models, and we will also discuss what lies ahead in this journey. Back to Martha.
Sorry, I was on mute. We will give the floor now to Mr. Mark Gambridge, the head of clinical investigation of MHRA, and Alexander McLaren, the benefit risk assessor on MHRA, who will give us an overview of why is important this pilot from the regulatory perspective. Go ahead, Mark.
Thank you, Martha. Next slide, please, Dan. So the first question, why is this pilot important from a regulatory perspective? Well, this is something, this type of approach in-silico is not written into any current UK regulation, and I know it is being considered for European regulation. And because of the constructs of our regulations in the UK, to make any changes is a very lengthy process, typically 2 years from end to end. So aside of any significant regulatory changes, what we needed to do was to look at our current regulation and see where in-silico actually fits within the regulation. And what we found is that the majority of our regulation talks around clinical data, the generation of clinical data, the provision of clinical data. So we can see a gap there. So what we can do in the meantime to bridge the gap between the delivery on new regulations or updates to current regulation is to provide some guidance on the use of in-silico data in applications for clinical investigations. I should also flag as well that as the UK sovereign regulator, we are responsible for Northern Ireland. However, the European regulations apply in Northern Ireland, so we have to implement both sets of regulation from an MHRA perspective. Next slide, please. So I've mentioned already, in terms of the pre-market requirements that are cited in our regulation, so it talks about clinical data, meaning the safety or performance information that is generated from the use of a device, and that should be derived from clinical investigations or other studies reported in scientific literature of a similar device for which equivalence to the device in question can be demonstrated, or the use of published or unpublished reports on other clinical experiences. So there is something that we need to fit into regulation number 2 regarding the use of in-silico data. And as I've pointed out there is no reference to any other sources of data such as in-silico. So that is a gap in the regulation that we are actively reviewing and pursuing. Next slide, please. So if we look at European guidance, so the MEDEVs are the guidance documents for European regulation. Again, there is no clear mention of in-silico data there. And again, it talks about clinical data, and where demonstration of conformity with the essential requirements is based on clinical data, it's not deemed appropriate. Adequate justification for any such exclusion has to be given, and it gives 4 specific points there. But again, it doesn't specifically use the word in-silico. Next slide, please. So just some points to consider that we're very clear that regulation must be challenged, not just in this area, but in lots of areas. And we are constantly updating our regulation, but that moves at a very slow pace, but we need to move to capture in silico. I've already mentioned that changes to regulation are lengthy and require consultation, and typical process end to end is around 2 years. We've got to be very clear with the guidance for manufacturers and for academia, and it's essential. And the guidance can be developed more quickly than the changes to the current regulation. So we also considered as well what types of study can leverage in silico data. So should they be first in human studies, proof of concept studies, or pivotal studies? So that's something that we would need to address in our guidance as well, in collaboration with industry. Another question, would in silico data negate the need for a formal clinical investigation, or would we see a hybrid type of study? I think reading around the subject, I think we're probably more likely to see a hybrid study. So if we look at a typical TAVI study in the UK, anything around 200 participants will be included in that study. I think if in silico data is leveraged, then potentially you could significantly reduce the number of participants. But we also have to consider as well the burden on these participants. So in the first year of being recruited and treated in the TAVI study, there is significant responsibility with that individual for follow-up, appointments, etc. And often patients are lost to follow-up in these studies. They will exit the study at their own leisure after the first year, but most TAVI studies can go on for at least 4 or 5 years. So it can be a significant burden for participants. In terms of market approval, so the example that's already been presented, which is a design iteration, we need to establish whether that would need some patients to be implanted with that new device, or whether the UK approved bodies would be happy to issue a new certificate based on that new information that's been generated from the in silico. I've also put there as well to note the complication of both EU and UK regulation, and both must be aligned as much as possible. And that's not just for ourselves, but also for industry as well, because industry is not just specific to the UK, it can be global. And it's really important to ensure that each manufacturer knows what the regulatory requirements are in each jurisdiction where they market their devices. And for me, I come at this from an ethical approach that we must not forget participants. A question that I've mentioned before is how we inform participants that a lot of data in the study that they may be participating in has been generated from in silico data, and what that actually means. I also stress as well in all clinical investigations that poor patient information equals poor recruitment, which equals no study. So you'd be surprised the number of studies that do fall down after a very early stage because they can't recruit participants. And sometimes that is due to the poor information that the participants are given. And I think overall, the one thing that we've learned from this project and other projects that MHRA has been involved in is that we have to be pragmatic, we have to be forward thinking, and we have to be reactive now rather than proactive in this ever-changing market in medical devices. I think that's my last slide. I think Alex is next. Oh, it's me still, sorry, apologies. So I just wanted to give you a quick overview of what MHRA actually does when they receive an application for a clinical investigation for a TAVI device. So once we receive a valid notification, we assign various assessors to review the application. That will include a technical expert like Alex that you'll hear from shortly. And because of the materials that are being used and that there will be a long-term implantable device, we'd have to consider the biocompatibility and toxicology effects. So we use our expert in that area. Statisticians to look at the statistical plan for the study, and our sterilization experts as well, because these devices come as sterile. And for every clinical investigation in the UK, we would always assign an external clinical expert for their opinion. So under current UK regulation for a TAVI study, we have 60 calendar days in which to provide an outcome, and that will be to approve or object. For Northern Ireland studies, it's 45 days under European regulation. However, we can add some additional time onto that, another 20 days where we consult with experts. And for a lot of TAVI, they are new and novel devices in terms of their design. So in this scenario, we will request a safety and performance report after a set number of participants have been implanted. So for example, if the study plan was to recruit and treat around 150, 200 participants, we would ask for a short safety and performance report after the first 5 participants have been recruited and treated. And that just limits exposure to a device that may be faulty, it may have some issues with the implantation in a human, bearing in mind that a lot of these studies, bench studies, are, the devices will be used with animals in the first instance. So it's an opportunity for the regulator to monitor the study in its early stages, and also for the manufacturer as well, and for the study sites. Next slide, please. Is this yours, Alex, now?
Yeah, I think that's mine, Mark. Thanks.
Good.
Thanks, Mark. Good afternoon, and yeah, thanks, Mark. I think you touched upon some of what I'm going to say, but I've been asked briefly to cover other aspects of a clinical trial application that I might pay or may pay particular attention to in my role capacity as an MHRA assessor. And this is outside of those general expectations for the requirements in the standards, the ISO standards. Ultimately, all of these points of interest relate back to identifying whether there are existing or potential hazards, and really what has been done to limit or remove this risk. This is not an exhaustive list by any means, and some of these overlap with one another, but perhaps a good place to start is an appreciation of the device's innovation and novelty. To what extent is this as a novel technology, and maybe it's the manufacturing process that is novel in the application. Another important question is there anything about the device that makes it unique? What is its USP, its advantages or disadvantages over rival devices? Why would a patient opt for an experimental device over a similarly tried and trusted device that had already been marketed for several years, say? And this is where an understanding of the innovation and USP is really useful, really needed. Next slide, please. Some other considerations include: what are the early stopping rules, and are there safety trigger thresholds in place? Do I have confidence that the applicant has an escalation pathway for device failures or unexpected harms? Is there anything extraordinary about the indications for the device, the population being treated, and the inclusion-exclusion criteria? Perhaps the device is treating an unmet need. Are the primary and secondary endpoints reasonable and justified in the context of the application? And how complex is the design of the device? Does its complexity lead to doubt about the long-term durability of the implant? How does the complexity affect the operation and human factors? Could this complexity lead to greater potential for user error during delivery and deployment of the TAVI in this instance? And do we need to tread more carefully, as Mark touched upon, particularly in the early pilot phase? Perhaps a much more controlled, slower rollout is justified. And what is the anticipated learning curve needed to operate the device effectively by the users? How best do we protect those patients receiving the device in the early phases? So hopefully, sped through that, but hopefully this gives you a flavour of some of the key areas that we may focus upon in an application.
Yes, I can talk a little bit on ethical considerations. For this pilot, there are two mainly punctual questions that we have discussed. One is, how can we ensure potential participants in a clinical investigation utilizing in-silico data can be informed to those participants? And the second question is, what good patient information look like? And this we consider that is very important from the regulatory point of view as well, right? Above the problematic, or because it's a problematic, the the considerations on data protection and on data privacy. As well, the patients have the right to be informed, and this may be raise an issue if they are not very well informed how their data or the health data is used, especially when we are talking about other uses from the regular human trial. But as well, we have global ethical considerations. So in the last RAPS convergence in Pittsburgh, a few weeks ago, we presented what we call the ethical considerations in a global perspective. There are two tiers where is the currently a two-tier adoption of policy for computational modeling and simulation. And when the USA, so America versus the world, are competing in the terms of establishing the rules for ethical considerations. Most of the rules are colliding or superposed with the rules of artificial intelligence ethical considerations. Then we talk about as well of risks associated to computational modeling and simulation based medical devices that are only being available with certain markets, and the risk that impose for manufacturers that may not adopt computational modeling and simulation. What are the risks associated with the low innovation path as well? And then there are lack of regulatory framework or international accepted standards. For example, in Europe, we don't have a medical device coordination group working yet, but we are working on convincing the MDCG to have a guidance for the EU. And also, as well, the other considerations in how the notified bodies in EU and in UK, how the approved bodies will be working, and we want to avoid that they work in silos and they can be communicated in order to have a kind of harmonized way of assess in silico evidence as well. And we move to the next slide, and moving as well to the next session, where we have Dr. Stephen Krauser, from Senior Manager Engineer from Exponent, Dr. Kehan McVeigh, that already presented, Dr. René Bombin, who is Associate Professor of Cardiac Surgery from QTech, and three of them will be presenting in the next slides. Go ahead, Steve.
All right, thanks, Martha, and thanks everybody for joining us and all the awesome information we've received to this point. Edan, if you could move to the next slide. So what we want to talk about here, and you didn't mention this in terms of setting the stage for much of the discussion. We're talking about a TAVI scenario. In this particular scenario, from a business perspective, there's a slight change to the device frame, and we are in a situation where we want to ideally consider reducing the size of a clinical trial. And of course, we want to use in silico evidence to try to support that based on all of the considerations and the information associated with the regulatory approval process and the approval process for starting a trial, as just discussed by our regulatory colleagues. This graphic here, there's a bunch of arrows, a bunch of things going on. On the top, we have in green the predicate device and the pathway that the predicate device took to get to approval, which in a very traditional sense was based on some bench testing, perhaps there were some animal studies, traditional clinical trial, unsupplemented by in silico evidence, leading to an approval process. So in our situation, that predicate device already on the market, doing a pretty good job. The company, as they should want to, seeks to make a slight change, perhaps, to improve the clinical performance of this device. And so they do this redesign to their device. Now we're in the bottom section here in the lilac color block, and we want to consider how do we land on the right-hand side of this purple block using both in silico clinical trial evidence as well as ideally a clinical trial with reduced participants. The goal here being leveraging the In-Silico device testing, the bench testing, and the validation between those, as well as the In-Silico clinical trial, again, to reduce the number of participants in a clinical trial. We're not talking about completely replacing the trial. We are talking about what we can do to reduce the size for all the important benefits that might bring, to ultimately get us to the point where ideally there is approval, assuming that the device meets the requirements that it has to have. So we want to get to this point, and we want to think about how do we consider all the various risks that are associated with these devices, and what can In-Silico methods do to assess those risks and give more confidence at the start of a trial, as well as with the information that might come out of an In-Silico clinical trial. So on the next slide, we've got a number of things, and I like this, and I think about this, and you can sort of see the arrows and see how this manifests as a stacked filter, where we go from the risks up at the top. These are a number of different risks associated with TAVI, all the way down to the bottom, where we have our prioritization for In-Silico assessments. And so we'll walk through these stages, but the basic concept here is that as devices look to move from the preclinical stages into clinical stages, there are a number of risks that need to be assessed. And according to risk management frameworks and assessment methods, various standards, we want to make sure that we have as much information as possible to assess the relative severity and the consequences of these risks using the information at hand. So when we lay out all the risks at the top here, and we think through the filters of the different stages to get down to the context of uses that we have on the bottom, we have a number of different reasons why certain risks may not be associated with or necessary to assess with In-Silico tools. The first one associated with regulatory relevance, which is a bit of a loaded term, but here we're thinking about to what extent is a particular risk something that might be flagged by a regulatory body. So as an example, on the right-hand side here, we're talking about a slight change to the frame. The frame material is not changing. So therefore, with materials being unchanged, it's likely that a biocompatibility assessment would be minimal in this circumstance. We certainly want to talk about it, but it might not be a full exhaustive study. So we think about those regulatory relevance situations and what makes it through there, what is still of concern. We get to the mechanistic linkage. These are circumstances where we want to think about the risks and think about whether the design change might actually have an effect on these risks. So something like hemolysis, transvalvular regurgitation, in our opinion, based on the recommended change or the suggested change to the device frame, we don't think there's going to be an assessment there. Now to the bottom one with the blue, In-Silico evidence value. This is a statement or a question that we want to ask in terms of whether or not we need to move to In-Silico evidence, or can we cover this on the bench entirely? So something like device migration we see there is caught in the filter of the In-Silico evidence value, because we feel that the testing methods, and Cathal will go through these in a second, are able to capture something like device migration. But the key point here is that we have 3 of these contexts of uses that make it all the way through these filters, if you will. So 3 risks that are not addressed or are still a concern working through these filters. And those 3 are unsuccessful deployment, conduction disturbance, and paravalvular leakage. Now I want to pause here. We're going to get to the questions of interest or the decision of interest in the context of use in a second. But I want to point out that as we're thinking through this, our framework is really assessing these risks. And we are, and I think this is a really exciting pilot specifically because we are considering multiple contexts of uses here. And we're considering how they might interact with each other. So on the next slide, Yidan, we have this framework. And the idea here is that we have this general question of interest. So that question mark OI, question of interest or decision of interest, where we're thinking about from the broad perspective, whether this minor device change is going to, you can read the text there, but whether we can reduce trial size with In-Silico evidence. That breaks down for us into 3 questions of interest and associated contexts of use. The first one is essentially deployment. The second one is associated with conduction disturbance. And the third one is associated with paravalvular leakage, or leakage of the blood around the valve, not going through the valve, but around the valve. With each of these, there are certainly related. And in particular, we feel that question of interest 1 is a derivative or a predicate rather for question of interest 2 and 3. We're going to think about deploying the device, taking a deployed simulation or a simulation of the deployed devices, and then assessing contact pressure in question of interest 2 and fluid simulation to look at leak magnitude and so forth in question of interest 3. So it's a situation where we have a workflow between question of interest 1 down to 2 and 3. Now, in considering this and thinking about where we might get information and how we might be able to validate and think through the risk assessment of these questions of interest, we put together a rather complicated table on the next slide. The key is we are thinking about all 3 of these contexts of uses, question of interest, associated with the 3 rows that are boxed here. There's quite a bit of information on here. There's quite a bit of assessments that we want to think about. But the key as we think through this is essentially for each of these 3 risks, so deployment, conduction disturbance, paravalvular leak, there are various test methods that are going to potentially help us to assess these risks. There is clinical information that we need to consider as we think about the risk assessment side of this. But all of these 3 are related to each other. Now we have different timescales. We have something we're referring to as domains, and you can read more about that in the paper when we publish it. But the basic idea here is we need to think about the testing and how appropriate the various testing methods are and whether there's testing methods for conduction disturbance, for instance, that you see here laid out in the standards. And then furthermore, what sort of information do we need to draw from clinical studies to make sure that our in-silico clinical trial is representative of the appropriate patient population? So with that, I'll kick it over to Cathal to talk through the bench testing, and then Renee will kick up on the clinical.
Okay, thanks, Steve. Okay, so Steve has introduced us to these 3 contexts of use. So we'll have to remember those. It's deployment, PVL, and conduction disturbance. And those will come up again and again as we get through the rest of this talk. And he's also kind of introduced us to the fact that there's this bench testing that happens. And how are they relevant to these three contexts of use? So I get to start with, why do we do testing at all? So in this scenario, the manufacturer would do testing for 3 distinct reasons, which are laid out here, 1, 2, and 3. Number 1, the new device will have design requirements just like any device. Those will need to be verified. That's a given. So we can set that aside. The second one is perhaps you would do comparative bench testing. That could be used to demonstrate that the new device is expected to perform equivalently or maybe even better than the predicate device. That head-to-head testing is invaluable. It could form a really important part of a submission and an important part of that justification that I talked about earlier to reduce the trial size. So in terms of the in-silico strategy, that's the whole point of this talk, that testing, and this is important, that testing could also act as an alternative source of evidence that is not modeling. And the modelers will know that if you have an alternative source of evidence that's not modeling, that reduces the overall model influence, which reduces the overall model risk. And the validation that we do of the model is commensurate with the model risk, so that'll have implications for how well the model needs to be validated. The third thing is the plain old model validation. We do testing to validate the models. We'll have models in this scenario, or in this theoretical pilot approach, we'll have models for the new and the predicate devices. Of course, if we're using models for an in-silico evidence strategy to do in-silico evidence modeling, then those models need to be validated versus comparator data. This is where the comparator data comes from. So those are 3 kind of distinct categories of why we would do modeling. If we go to the next slide, we can dig in a little bit more to how we know what type of modeling to do. So again, we go back to the standard 5840. It's kind of our guiding light through these things when it comes to the type of testing that we do. Again, I've already said this, but again, it helps us with how to determine what bench testing, animal studies, and clinical studies need to be done. So we'll dig into that a little bit more in the next few slides, but we'll really focus on the testing that's relevant to those 3 contexts of use. Again, deployed shape, PVL, and conduction disturbance. So let's just skip to the next slide, please. Top left, I've just highlighted the context of use that the slide's relevant to. So first one up is the deployed shape. The standard, it says that I'll just read some text on the right-hand side there, that the intended performance shall include an ability to conform or interact with anatomical structures. Of course, that needs an understanding of the deployed shape. There's no, detailed or prescribed testing in the standard for deployed shape. It doesn't tell you how to test the deployed shape of the device. But we do know that there is out there good, well-accepted testing approaches to characterizing what is the deployed shape of the device. I've given the example here of micro CT, which is pretty accurate these days and can be used for that. Next slide, please. Radial force testing, that is discussed in the standard pretty extensively, and it also says that high frame radial force, it's given as an example of a hazard that's mechanistically related to conduction disturbance. So that links radial force to conduction disturbance. So the good news is we do have a test for radial force, and I mean, you could theorize that local radial force may be a predictor for conduction disturbance. And that's important because there isn't a test described or prescribed in ISO 5840 to directly address conduction disturbance or to measure contact pressure. If we go to the next slide, we'll talk a little bit more about conduction disturbance. It's only really discussed in the standard in relation to preclinical animal testing and in the clinical assessment section. But again, it's important, and Steve also mentioned this, there's no guidance in the standard that tells us how to directly test for conduction disturbance. And I mean, as far as I know, there's no accepted way to measure things like contact pressure and be able to predict conduction disturbance on the bench. Next slide, please. So on to PVL. PVL testing is, again, covered very extensively in the standard under the hydrodynamic testing section, with standardized test conditions, tabulated and called out, and also challenge conditions that I've highlighted here, where there's a nodule with certain dimensions. It really mimics calcification of the anatomy, and the challenges that poses for PVL. So that's really a challenge condition. So summary here is that there's really good guidance in the standard around PVL testing. So that's my last slide. I'll say in summary, as I mentioned, the testing serves three purposes. Design verification, obviously we've got to do that. Head-to-head testing, if we're comparing to a predicate device, that could be done. And validation of our models. I covered some of the standardized tests in 5840. They are standardized and they can be used for that purpose, and they're very important for that purpose. Having said that, the endpoints that we're talking about here, PVL and conduction disturbance, they're very driven by the interaction of the device with the patient's anatomy, which is far from standardized. And of course, it's highly variable across all the patients that we treat. I'd say that variability, it's naturally baked into clinical studies. When we do clinical studies on the bench, it is very impractical and prohibitive maybe to try to cover that level of variability that we would expect to see in patients. That's a different task than running standardized tests. And for things like conduction disturbance, again, it may not even be possible to do that on the bench. So that's a huge motivation for In-Silico approaches. The beauty of In-Silico approaches is that it enables us to cover that whole spectrum of anatomies virtually and to simulate them. And, it allows us to interrogate those like detailed local interactions, things like contact pressure between the device and the anatomy that drives things like conduction disturbance and PVL. So for more on the clinical aspects of that, I'll hand it over to Rene.
Thank you so much. Yes, it's great to be here, and I would like quickly speak about the clinical aspects for conduction disturbance and also the paravalvular leakage. If we look into that 30 days post transcatheter aortic valve replacement, there's still a risk. I mean, even after now many years that this technology is on the market, there's still the risk for pacemaker implantation. And it's a common risk. Recently published in 25, we had like a meta-analysis with over 100,000 patients. And I'm going to present just, that you get like a feeling of what this actually means with regard to clinical aspects. And as Kahal said, there is actually no possibility to test anything on the bench with regard to the conduction disturbance. But I think we have to look into the risk factors that we have actually there to consider. So things that can happen, like a right bundle branch block that is already there. So that's a very strong risk for a pacemaker implantation after TAVI. Then an AV block, right? First degree AV block before TAVI implantation. It's also, here we have a moderate risk for left bundle branch block. It's actually not significantly associated in this meta-analysis with then pacemaker implantation after TAVI. I think one important thing to say is that there are procedural risks also. So it's not only what the patients actually bring into the operation, it's also the procedural risks here on the left-hand side of the slide. It's actually the implantation depth. I mean, the farther you go into implantation site, it might increase, or it will also increase risk for pacemaker implantation. Also the valve type. Self-expanding valves also increase significantly the risk, and a balloon predilatation. So everything that actually we know that the pressure that we bring into the aortic annulus, that this is interfering actually with the risk for pacemaker implantation. And we see here an overall 30-day pacemaker implantable or necessity is around 70.5%. And I mean, this is still pretty high. Next slide, please, Kahal. Okay, so what do we have here on this slide? I said, I mean, again, I mean, it says that there is still not every patient who is actually diagnosed or who need actually a pacemaker immediately after the implantation also remains pacemaker dependent after 12 months. So it's only a part of these patients. So not every one of these 17% will then also remain pacemaker dependent after a year. However, still, it is still high. I mean, the range is, so it is a risk that patients actually need a pacemaker after this procedure. What we have to consider is the long-term association with pacemaker dependency and all-cause mortality, because pacemaker dependency means that if you, I mean, depending on how you pace, but I mean, the permanent right ventricular pacing will then more or less lead into a heart failure with more rehospitalization. So this is something that we must consider definitely. So next slide, please. Here we'll talk about paravalvular leakage. As it was explained before, paravalvular leakage is actually the blood that actually goes not in the middle through the valve where it should go. So it's actually what is going on the left or on the side of the valve. And as Kahal said before, so it's in diastole. So it actually puts load on the ventricle in the phase where the ventricle should relax anyway, so in diastole. So there is a constant pressure on the left ventricle. That means if we have paravalvular leakage is graded, and we have different types of paravalvular leakage more than 2 is actually also associated with a higher mortality after 6.5 years with a higher mortality risk. So this is something that we must definitely consider. So having a paravalvular leak means that maybe due to anatomical circumstances like calcifications or maybe not a perfectly round annulus, that means we need maybe higher pressure to put the valve or to align the valve with the anatomical surrounding. But this would mean a higher pressure to reduce the risk of paravalvular leakage might then be more associated with a higher risk of pacemaker implantation. So that means reducing paravalvular leakage. I mean, one of the topics that we have here in our pilot is not just nice to have, but it is a procedural priority. So it is important that we cut down on paravalvular leakage as far as we can. Okay, that was my part here. Thank you.
Yes, thank you so much to the 3 presenters. We go to the next session, that is the risk assessment and in silico credibility evidence presented by Dr. Alisa Rami, Dr. Pawan Kumar Pandey, and Dr. Yidan Shue. Go ahead, please.
Thank you, Martha. As mentioned, one of the core principles of the ASME VMV40 is that credibility is risk-informed, basically, which means that we do not apply the same level of verification, validation, or uncertainty quantification to every model, And instead, we require evidence that will be proportional to the potential clinical harm if the model goes wrong. So according to the ASME VMV40 and FDA guidance for credibility assessment of the CMS models, the risk defined based on two main contributors. One is the decision consequence, which reflects the impact of the incorrect model, basically, and model influence, which reflects how much the regulatory or clinical decision depends on the model. So as Kahal and Steve and Rene mentioned, so we looked into the clinical environment, we looked into the clinical literature around TAVI to basically study different harms around TAVI for each of the contexts of use that we are going to focus on. And we also looked into ISO standards to reflect on severity for different harms. So severity will range from catastrophic or fatal outcomes through the minor to negligible effects, and frequency around TAVI start from like frequent, probable, occasional, remote, or improbable. So for the context of use in this regulatory scenario, we think that the deployment is high consequence, because if the deployment model is inaccurate, it can lead to malapposition, embolization, coronary obstruction, and even stroke, which are high consequence. Conduction disturbance, as mentioned by Rene, is again high because of the consequences, and paravalvular leakage because, so if you look at the occurrence rate, and because the PVL is usually managed electively rather than emergently, we can say that the consequence is medium for this one. And if you want to study the model influence based on what Steve mentioned about this pilot scenario, here we are going to have like supporting evidence from an in-silico trial, which is what we are designing. In-silico trial of the predicate device, the clinical data from the predicate device, reduced size clinical trial for the modified device. So we think that across all the three contexts of use, we have a medium model influence. And if we combine all these two, for the device deployment and for the conduction disturbance context of use, we have medium to high overall model risk. And for the PVL, we are going to have medium model risk. So these model risks are going to drive the credibility activities that we are going to do later, and my colleague Pawan are going to present.
Now that we have the risk assessment, to develop the trust in the in-silico trials, it is necessary to have some credibility evidence. And thankfully, FDA has provided some guidance on different categories of these credibility evidence that can be presented. So there are 8 different categories, and I will discuss these 8 different categories of credibility evidence for the 3 different contexts of use that we have presented previously in the next slide. So the first category is the software code verification, which is essentially the fact to check whether whether the software accurately converts or translates the mathematical equations into the code or the numerical algorithm properly. This verification is often done by the software companies if you are using the proprietary software, but at the user's end, it is also necessary to verify the benchmark results for your current installation version and the hardware that you are using. The next step comes in terms of the model calibration, where we fine-tune or decide the material properties or the parameters that can govern the system behavior for different kind of context of use. For, let's say, the deployment and the PVL, it will be the material properties of the device and surrounding tissues. For the conduction disturbance, it can be some surrogate markers, threshold values, such as the stress and forces which exert on the surrounding tissues. And for the then the third category is the bench test, which is essentially where we validate our simulation results with the experimental results that can be obtained in the lab. So for the deployment, conduction disturbance, and paravalvular leakage, we do some basic experiments in the lab and we match those results to do the bench testing. Then the next two categories are the in vivo validation and the population-based validation. These are basically validation of the simulation results with the patient-specific results. But in the in vivo validation category, we are able to compare results by doing the one-by-one comparison between the same patient's anatomy. But in the population-based simulation validation results, we cannot do that because the exact patient cohort may not be the same, but they can be comparable. In those situations, we try to do we try to show a statistical equivalence between the physical quantities that are being calculated through the simulations, and we try to match them with the study endpoint of these clinical trial results. Then there are three other categories which may not exactly fall into the verification or validation type of credibility evidence, but they provide some extra level of assurance of the correctness of the prediction of the in silico trials. For example, the emergent model behavior can be there, although it cannot be applicable to all of the context of use. But let's say for the deployment, it may be used to predict or identify in some cases the buckling or the infolding behavior of the stent. Then there can be a model plausibility kind of evidence where we visualize the final deployed TAVI geometry or the flow structures that form because of the PVL. And then there is this last category where we do the uncertainty quantification or the calculation verification by doing the mesh convergence study on the new device, or we try to identify the maximum contact pressure or similar quantities, let's say, that satisfy a certain threshold value that is defined for that particular context of use. Now that we have identified three different contexts of use, we have a certain risk assessment, and we have a fair idea of the credibility evidence. We come up with this validation plan that will be applicable to the all three contexts of use that are being developed, that are being discussed here. The key thing to note here is that there is a slight change in the device in all three contexts of use. So as you can see here, there are several boxes in this schematic. The green box represent the activity that is being done to the predicate device, and these purple boxes are the activities which are being done with the new device. So the key thing here is that we first do the bench test comparison to lower the model influence. Then we do some simulations on the patient's cohort and some geometries, and then we compare these results with the bench test results which are obtained for the new design and the predicate design as well. But this clinical trial, the in silico trial that we do here, is being done with the predicate device, so that we can, because there is a relatively close relation between the predicate device geometry and the new device geometry. And then this clinical simulated clinical trial results are used to reduce the number of participants that are required for doing a clinical trial for this new design. So now I will hand over it to Idan, who will discuss the credibility requirements.
Thank you, Pawan. So here is a brief summary of what we have discussed. So we have determined that for context views 1 and 2, the model risk is medium high. And for context view 3, the model risk is medium. And I also list the 8 evidence category from FDA, Pawan has just mentioned. And today we will focus on discussing the credibility requirement for the in vivo validation and population-based validation, which are unique for this parallel study. So basically, we all agree that it is a manufacturer's role to determine the credibility level required corresponding to their model risk for their in silico regulatory submission. But here we just formulate some recommendations for what manufacturers can actually do in their practice. So if we look at the credibility factors and goals in ASME VV40 and FDA guidance, actually there's no written factors and goals for every scenario, but we can have some general understanding from this recommendation based on a centrifugal blood pump example showing on the left. And for our model risk, that two are medium high and one is medium, we require the manufacturer to reach at least level 4 out of 5 for conduction disturbance and deployment, and at least 3 out of 5 for the parameter leakage. And based on this recommendation on the left for the blood pump, the difference between the simulation results and the experimental data should be less than 5% error between each other. And the manufacturer should also consider the uncertainty of different model components and how they propagate through the workflow. And so firstly, we look at the in vivo validation results. And in this case, it's, as Paul mentioned, it's head-to-head comparison of study endpoints between the patient-specific modeling and the clinical outcomes from the predicate trial dataset. And the three things we need to look at are deployment geometry, conduction disturbance, and probability leakage. And for the uncertainty, there are several categories need to be considered. Here, we just summarize the three most important ones, which are the model representation, consisting of device geometry, and the virtual patient, which are a digital shadow cohort geometry. And the second one is model form, where we need to consider the uncertainty in the tissue and device material model. For example, on the right, I'm showing you the some models used for aortic tissue. Like you can see, there are tons of models out there, like from the papers. Some people assume they are rigid, some assume are linear, some use different nonlinear model for them. And you can see they give different results when you use different models. And in addition, like when we assess the PVL, we need to consider the fluid flow rate waveform, and also the fluid viscosity models, and so on. And we also need to consider the model parameters in all the model forms we consider in this case. And for population-based validation results, like we suggest, the manufacturer need to look at two study endpoints when they compare their cohort-based outcome from the simulation cohort to the clinical dataset cohort. The first one is the occurrence rate of post-TAVI conduction disturbance, and it's recommended the manufacturer can look at different types of conduction disturbance following TAVI, for example, LBB, AV3B, or like even AV2B2 and other types. And secondly, the manufacturers need to look at the occurrence rate of different levels of paravalvular leakage following the TAVI treatment. So like there are usually 4 categories, which are no, mild, moderate, and severe. And also the manufacturer needs to consider the uncertainty in their population-based model, which should inform the sample size they chose for their virtual patient cohort to ensure their results are statistically significant. And we also highlight the point that it's manufacturer's responsibility to justify that the chosen sample size has sufficient power in their analysis, but we can get some information on the order of magnitude of the in-silico patients we need from some existing standard. For example, from this ISO 5842, we can see that for each valve position, the manufacturers require at least 150 patients in their clinical trial, and they need to follow up for 1 year. And also they need to consider the uncertainty, like using standard statistical methods to make sure that their sample size has sufficient statistical power. But in some cases, the manufacturer might have some issue that their chosen cohort doesn't have sufficient digital shadows for their in-silico clinical trials, or they want to enhance a certain underrepresented population in their trial. So they might need to consider the use of synthetic virtual population models, either statistically based or machine learning based. And the credibility of these models has been covered in the UK CEiRSI Panel 3, which is available on our YouTube channel. And you can find more information there if you're interested.
Thank you so much, Yidan. Now we pass to the next section, and I will invite Dr. René Bombin to talk about the importance of the predicate clinical trials.
Thank you, Marta. Next slide, please. why do we actually need to look at predicate clinical trials in this case? So when we try to establish now the credibility for whatever we are looking for, I mean, we should also look into what is already published. So predicate clinical trials for TAVI, I mean, one thing that was established here was actually safety or data for safety and performance or efficacy, so that we actually know what we can expect, and also benchmark performance if we have now next generation iterations for our device. And I think in the TAVI field, a lot of studies has been done. I mean, we will see in the next slide some of the major studies that has been used actually also for regulatory approval over the last 15 years. I mean, starting here in 2010. So we have successfully having different types of studies. I mean, all big manufacturers that actually produce these valves also initiated trials. So the results of these predicate clinical trials are important now also to build the basis actually to compare our model results with. And next slide, please. If you look into the importance of predicate clinical trials, so what we actually need this for is actually to have like a good picture, so that we know that that we have cornerstones in the field of TAVI studies. We established with the predicate clinical trials, as we said, the safety and efficacy benchmarks. And also when we now go into new devices, that we have a basis for this innovation. What we always have to do, and I think this is clear, that we continually have to update these predicates to ensure that also the data that we have for the cornerstones for safety and efficacy, that they will stay the same. However, also these predicate clinical trials that we're looking into, they also have risks, or they come with risks and limitations. The over-reliance on predicate data that we have. So we also have to see if the data that we know for paravalvular leakage and pacemaker need, they might also differ. So we also have to see this with a certain amount of cautiousness. Then we see that we have evolving populations, right? I mean, back in the days, only high-risk patients have been treated. Nowadays, there are also big studies ongoing that go more into low-risk patients, where actually the risk for surgery, for open surgery, is then even higher, considering or comparing with a TAVI procedure. And then, of course, the procedural learning curves and the device refinements. I mean, looking here into the three aspects of our context of use, when we say we look into the deployment of the device iteration, we look at the contact pressure that we're going to use, and also then that will result, of course, into paravalvular leakage or the risk of pacemaker implantation. So we have to also consider these procedural learnings when we look into the data that are already there from our predicate clinical trials. Thank you. Thank
you, René. We will give the floor to Dr. Adesola Demiloye to talk about the interaction and impact of artificial intelligence and computational modeling and simulation. Go ahead, Adel.
Hi, everyone. Thank you so much for joining us today. My name is Adesola Demiloye from Swansea University. In this section, I will be talking about the role of artificial intelligence in computational modeling and simulation. Basically, we want to see how we can use AI to enhance medical procedure simulation. Next slide, please. So what do we know at the moment about computational modeling in TAVI in terms of the limitation, and how could AI help in addressing some of those limitations? In simulating the implantation of TAVI devices, we know that there is a lot of computational cost. This could be in terms of the journey from point of insertion, or even to the deployment of the device itself. And so this requires a lot of computational resources in terms of mesh generation, in terms of assigning material properties, boundary conditions, and so on. So that's one of the main limitations with computational modeling of TAVI deployment. The other part, which has been mentioned briefly as well, is in terms of patient variability. So for different patients, you have different anatomy, different specific conditions. And by having this variability in the patient's condition, it also impacts the computational modeling of the TAVI device deployment. So it is very important to consider the anatomy variation, the hemodynamics, and this could also impact the perivalvular leak of after deployment, after a couple of months, years, and so on. So the last part of the consideration in terms of limitation is the issue with the lack of integrated workflow. When we use computational modeling currently, or in most cases for TAVI implantation, we consider it as a silo. So we just simulate the deployment, we simulate the different aspects individually, but it is very important to have a workflow where the systems and the different stages talk to each other. And the question then becomes, how can AI help in addressing some of these limitations? In terms of computational time or computational cost, we could explore the use of surrogate modeling or even reduce order modeling. So in this case, rather than simulating the entire process repeatedly for different patients, different cycle, we could actually use the generated computer simulation result to train surrogate models that can then be used to improve the speed of subsequent simulation. And here, in terms of the frame deployment, we can use CNN, convolutional neural network, to actually achieve this purpose. Then when it comes to stress distribution, there are other AI-based algorithms like the graph neural networks that can be used to predict the vomitive stress on the aortic wall, and by so doing, we are able to improve the speed of our computational simulation. And of course, at the same time, we can use AI-based algorithm like the physics-informed neural networks, and that could really also help to improve speed. The other aspect when it comes to patient variability is the use of AI to generate the geometry. So in terms of segmenting the patient-specific images, and also in terms of mesh generation. Because again, if you have to generate, perform segmentation for thousands of images, because in recent time we can generate a lot of images for patients, but analysis of this image can take images can take a long time. So with the use of AI, we are able to automate the segmentation. For example, you can use the UNet or the NN UNet algorithm or architecture to speed up the generation of to segment the image from our CT or our MRI data. Then in terms of the mesh generation, we now have what we call the generative models that can actually create patient-specific meshes, and this can also be done real time, such that we can also capture the specific changes in the patient's anatomy. The last part of this AI application that I would want to mention is in the area of the FSI coupling and a real-time adaptation. So in the case where we have to consider different aspects of the TAVI deployment or even the long-term use of TAVI, it is very important that we have a system that where this structural element or the structural mechanics is communicating with the fluid mechanics in a seamless way. And so to achieve that and to minimize, of course, computational cost of passing information from one part of this system to another, we could actually use AI or the recurrent neural network where certain information are saved and then passed on to the next stage without needing to recompute to run the simulation over again. And this can be used to predict the pressure gradient and also to compute what we call the wall shear stress distribution. And very importantly, we can get the paravalvular gap formation and leakage severity as this real time within our simulation. In the past, there have been some successes. On the next slide, please. We had some successes with industry. For example, AI predictive modeling was recently approved in 2023 by the FDA for DASI simulation. Here, AI-based system was created, which they call the precision TAVI, where they can use AI to carry out individual patient-specific computer modeling of the TAVI procedure. And this can, according to the manufacturer, can help to predict the patient outcome and also enhance the pre-operation planning process. The other aspect of the current industry application is in 3D image reconstruction, and even in terms of having a workflow that can be used pre-operation. So this was an example of this is the Siemens SIGO Aortic valve guide. And also we have very recently the merger between or the acquisition of FEOps by Materialize, and this can be seen within the Mimics Planner software, where you can actually plan the surgical process within a software environment. These are all steps towards having an integrated workflow. However, there are still a lot that needs to be done. So there are challenges ahead. And some of the challenges ahead in using AI when it comes to TAVI or improving patient outcome is in terms of data scarcity. So despite the fact that you have a lot of images for each patient, these images are sometimes not all annotated. And in that case, it makes it also difficult in training your AI model or even in deploying within the AI system. Then we also know that in order for you to be able to use the AI within a regulatory framework, for example, it needs rigorous validation. So it has to be validated against several clinical outcomes. Of course, we know that TAVI is almost 10 years old now. So there are long-term data that are coming across now. And hopefully within the next couple of years, with further research, we will be able to create a rigorously validated AI model based on the past data from the current deployment. Then the other challenge that we have is in terms of the black box nature of AI, as is mainly defined, in the sense that you cannot tell what is really happening within the AI software. So, and that has really led to a lot of challenges or skepticism from clinicians in terms of adopting this AI. So essentially, we need to get to a point where we are able to validate rigorously the AI model and then provide clear information on how this has been validated for possible clinical application. Then the last point there is in terms of generalization. So the question here is, if you train your device using a certain set of data, could someone else use the trained model within their own setup? So we need to be able to confirm and say, yes, like a physics-based model, when you deploy this model, you get the same result as long as you are using a consistent parameter. But then with AI, there's still that issue with generalization. So those are some of the challenges that needs to be resolved. Of course, looking forward, there is a future to the use of AI when it comes to TAVI deployment and simulation. And on the next slide here, we are looking at a possibility of having an automated workflow where you can use AI to improve decision making. For example, you could set up an agentic simulation where there are different agents that talk to each other, and you set a very high level objectives. For example, you can say, okay, within this, for this particular patient, minimize the paravalvular leakage and also avoid conduction disturbance. That is a high level objective. And then provide the patient data, provide this CT scan, the MRI images, provides some results from existing benchmark tests, and then let the AI simulation or the simulator carry out the optimization and return the results. But not just return the result, it should also be able to return the results in a way that the results obtained are explainable. So the AI should be able to then explain how certain decisions were made, why certain decisions were made, and by so doing, we can be able to trace the decision-making pathway, and that could also help to enhance the adoption of the AI-based system. So in conclusion, it's very important to understand that there will be trade-off. There will be a lot more work needs to be done, especially in terms of the long-term assessment of TAVI using AI and AI-enhanced computational modeling. Next slide. Okay, and that is it from me. Thank you.
Thank you. Thank you so much. Now we will go with Stephen, back to Stephen, in order to give the final conclusions of all our discussions. Stephen, go ahead.
All right, let's land this thing. So concluding thoughts. First, before it's not even on these slides, I do want to thank all of the participants in these discussions. We have had a lot of conversations, a lot of very good conversations. And I can't speak for everybody else, but I'm certainly excited to have shared our thoughts during today's discussion. Look forward to the Q&A. So definitely hats off to everybody, including Martha, for being a wonderful organizer and leader on this project. So thank you, Martha. Just with respect to concluding thoughts, I think one thing that's really interesting about this one, and I was really excited about how this evolved, specifically because this was a realistic scenario. Cahal showed the slide earlier with the evolution of at least two of the premier TAVI frames. And you'll note in that, and he mentioned the idea that there are slight modifications throughout the life cycle of a product. This is a very realistic situation that we were looking at. And it was one that I really appreciate because while we were thinking about an in-silico clinical trial, it wasn't from an efficacy standpoint necessarily. It was more importantly from the perspective of interrelated risks. And hats off to the clinicians and those representing the regulatory side of things in these discussions, because one of the key considerations that that really came out of our early discussions and drove much of this work, if not all of this work, is the fact that when we're thinking about risk, you can't really think about risks in isolation. It's really important for us to be thinking about how these things connect together. In other words, if you're going to think about PVL, that's great, but you should probably also be thinking about conduction disturbance because, as the team laid out, they are related to each other. They are related to how the device is deployed and so forth. So I think this particular pilot and the team and the discussions was really valuable because we did talk about these multiple questions of interest and context of uses, the fact that they tie together, and the importance of considering how those tie together when you are considering risks, which is obviously very valuable from the perspective of in-silico clinical trials. So really love this group and love the conversations that we've had and look forward to carrying these forward, both through the paper that we mentioned as well as some of the documents and so forth that we'll put through the CEiRSI project as a whole. Some important limitations. So Mark wants to make sure and repeatedly emphasize the importance here, which we all agree with, of the delivery system and any potential effect that the delivery system might have on this device. Part of our first context of use was associated with the deployment of the device into simulated aortic roots. And that's obviously very important. The delivery system plays a huge role in that. And we did not look at that explicitly here, but something that should be noted and should be part of the overall matrix of considerations that one might have. The other thing I want to note here is just the importance of, and Mark and Alexander did lay out from MHRA perspective their considerations. It's of course important to consider the perspective of approved bodies, notified bodies, and other regulatory agencies in these discussions. We had some involvement early, but I think it's important that we consider all of our thoughts here through that lens. Of course, nothing that we said here is any kind of formal recommendation or guidance or what have you. So all of those things should come up through appropriate conversations with the appropriate regulatory agencies as you think about taking these methods forward. But with that said, on the next slide, we got a couple of figures holding up Q's and A's. So we would like to open it up to discussion here. Again, thanks to the panelists. Thanks to everybody for coming out today. Look forward to tackling some of the questions, although hopefully somebody else can tackle the first one, because that seems like a complicated one.
Yeah, yes, we have a question that I think that the best person to answer is René, because it's a clinical question. And it is like that. Why should a new generation valve be developed, since the risks for paravalvular leak and conduction disturbances is well known? René.
Thank you, Marta, for this question. And I think that's a good question, and I think also something that needs to be considered, because developing a new device, I mean, everybody knows this, right? It costs a lot of money, and is kind of also a risk, also with regard to the acceptance. However, I think with our pilot, that's what we are doing here. I think we are tackling two problems that has actually not been evaluated, because I mean, you can't in vivo measure the pressure that you actually apply to the aortic annulus. So this is why it is important to look into this, to look into, in this case, not measurable pressures that actually can then also improve the outcome for the patient. As we saw, yes, of course, it is manageable. I mean, if somebody has conduction disturbance, it might not go away totally, and you can implant a pacemaker, and if you have a paravalvular leak, you can somehow manage. However, you saw this meta-analysis with over 100,000 patients. Paravalvular leakage is still in the late course of this disease, still a changer for mortality, so a high risk for higher mortality. And as I also said, pacing also means that, yes, for the moment it's okay, but if you have a long-term right ventricular pacing in a patient with a heart valve that is already at risk for a structural heart disease, might then end up also with a higher risk. So knowing this, I think with this pilot is important to, if we now think about developing a new valve, we can actually tackle these two issues that has not been in the focus from a design perspective yet. And this is why I think it is important to develop a new valve. Thank you, Matt. I hope I could answer your question.
Thank you. Yes, totally answered. Let's go to another question. And this, I think that Ali can address, is about the risk assessment. And how would you decide where additional VMB or UQ would actually reduce decision risk in an in silico trial for a medical device like a TAVI?
Thanks, Martha. So basically, as I mentioned, so it's based on the ASME VM40 and the FDA guidance. All the credibility activities are basically risk informed. So we need to do first a good evaluation of the risk around the context of use, around the question of interest that we are going to address. So and for that, so we have the ASME VM40. ASME VM40 is largely based on like bench testing or single model testing, but we have also the FDA guidance for credibility assessments of the computational model and simulations. And also we have the publications from Jeff Bischoff and Fav, they looked into clinical validation. So we basically look into model influence in terms of scope and coverage of the influence of the in silico. So how much we are going to rely on in silico evidence solely, and then the decision consequence. And with these two combined, we give a good representation of the model risk. And this is basically the model risk that will finally determine the level of verification and validation that we are going to require. So we are more interested in a credible model rather than verification and validation in their theoretical definition.
Thank you so much, Ali. I have another question on animal experiment and the three famous R's. So first question, and I think that Jidan can answer these questions, is, in your opinion, Jidan, what is the role of in silico methods in replacing, reducing, and refining the use of animal experiments? And then how to ensure the use of model-informed evidence doesn't unintentionally bias patient selection and under-detected severe adverse events. And then I have two more questions related to that, but if you can start with that.
Okay, thanks, Martha. Yeah, actually, that's 4 questions. So I will first answer the first one on animal trials. So I think the answer is certainly yes. Like the in-silico methods can play a key role in reducing, refining, and replacing existing animal trials. For example, maybe not a TAVI example, but for a flow diverter example from our own group that we ran the FD-PASS trial for the assessment of a flow diverter device for treating cerebral aneurysm. And from that study, we have shown that like using this in-silico method instead of like going to animal studies, you can save at least hundreds of rabbits and dogs in usual development of these devices. So, and also, by the way, I also think the in-silico methods can provide like additional evidence, like more realistic evidence than animal models, because there's a huge physiological difference between the animals and the human. So for the second question, sorry, could you?
How to ensure the use of model-informed evidence doesn't unintentionally bias patient selection and under-detect severe adverse effects?
This is a very good one. So I think the first point is that we need to, like as we have mentioned in this pilot study, we need to have sufficient in-silico virtual patients in the in-silico trials to ensure that the results have a sufficient statistical power. And secondly, I think it's useful to use some new technologies to cover the patient groups that are underrepresented in existing trials. For example, like that's an example, like the RAPID-3 trial for Lotus Ball that was done in 3 continents, but was not done in South America, not done in Africa, not done in Asia. So like the people from these continents are underrepresented. So like if we can use some in-silico evidence, like patient-specific models or even generative models of like, for example, Asian patients, we can prove that like this device is also safe and efficacy, like efficient in the Asian population. Yeah. So that's
yes. And how to consider the interplay between conduction disturbance and paravalvular leakage, since both of them are related to the radial force and valve anchoring?
This is a very good point. I think like Renee has covered it very well in the talk. Yeah, just to mention like if you plant too deep, that you have a higher chance of conduction disturbance. But if you place too high, you have a chance of paravalvular leak. You also might have other issues we didn't consider in this pilot. For example, you might have some issues for accessing coronary artery for treating coronary obstruction later on. So like there's an interplay between, but I think as Stephen mentioned, like we need to have a connected risk assessment framework and considering everything in one instead of separately.
Yes. From all applications mentioned in this pilot, what will be the biggest opportunity for the use of a TAVI in silico trials?
Maybe you would like to answer this question?
The use of AI in the TAVI silico trials, yes.
Maybe you want to answer this question?
Yes, I can answer these questions. Yeah, so I think that is a big opportunity. What we are seeing normally is that the vendors, the providers of in silico platforms already are incorporating AI, any type of AI and AI models that serve in order to produce a computational model and simulation. So I see that there is a huge potential to more and more incorporate AI, especially agentic AI, and there are various big players in the market with doing already experiments and trials with the agentic AI. And that's why we cannot, as manufacturers, we cannot leave the opportunity go because are really good tools that can give us opportunity to make innovation in a faster pace, and also reducing costs in human clinical trials and animal clinical trials as well. Good. I have another question from Tosif Salim. Can the presenters comment more on the prediction of conduction disturbance and the extent of a prediction, for example, left bundle branch block or a complete heart block? Will it involve also anatomical features of the conduction system of a patient-specific level? From which imaging modality will this patient-specific data be obtained, for example, from a CT scanner? Will you consider patients' baseline conduction system disturbances? Who would like to answer this?
I have some thoughts on this topic. I don't know if it answers the question. It's probably between conduction disturbance and PVL. If somebody asked me to model both of them, I'd prefer to try to model PVL. Conduction disturbance is really hard to model.
Mm-hmm.
And it gets to a bigger challenge with in-silico clinical trials of there are things that we can model and there are clinical endpoints, and there's often a gap between those two things. With conduction disturbance, we can model things like forces and contact pressures. Are they predictive or indicative of onset of conduction disturbance that requires pacemaker implantation? We need to be able to bridge between that engineering prediction and the clinical performance of the device. That's still a tough problem. I think part of that is addressed with the approach we outlined here, which is you can do a lot with essentially comparative analysis. If you look at how your predicate device performed, and you can leverage the good clinical performance of that device, maybe it has an acceptable pacemaker implantation rate, and you can compare those engineering metrics from the new device back to the predicate device.
Mm-hmm.
Let's say it's contact pressure, for example. It might be other things like contact area, how much of the membranous septum are you actually in contact with. Then from that comparative assessment, you can potentially infer what the performance of the new device will be in terms of conduction disturbance. I think that'll get us part of the way there. Eventually, we'll have to close that gap properly and have models that can actually directly predict conduction disturbance. That gets into very complex multi-physics and physiology models that are a ways off. I'll try to answer the actual question. Yeah, generally these come from CT scans. CT scans can give us a pretty good picture of the anatomy. Yeah. And from that, we can extract out where is the membranous septum, where is the conduction system relative to where the device would be implanted. One more thing I'll say is, I think it was Eden mentioned it there, the depth of implant is really important here when it comes to conduction. And I had mentioned when I was talking earlier that the beauty of in-silico clinical trials is that we can look at a range of anatomies. Another really important factor is we can also look at a range of implanter behavior, if you want to call it that, or procedural parameters. What is the depth of implant? How is a device clocked? So you kill many birds with one stone. You can simulate one virtual patient, but you can also look at a range of procedural parameters for that patient, which is really useful when it comes to things like conduction disturbance. Yeah.
Good, thank you. Another question here from Visha Piet, if I pronounce well. How can automatic cementation and mesh creation from generative models enhance the existing planning process and simulation fidelity? And how much can we rely on those results, especially in cases of severe stenosis? I think that maybe Adesola would like to respond on this question.
Yeah, thanks a lot for that question. I mean, it's a very interesting and important question. I think you got me thinking again about some of the points I've made. But I think in terms of automated segmentation, one of the benefits will be speed. For example, like I mentioned earlier, we have, for a particular patient, you have a lot of images that you're generating or you capturing from the CT or even the MRI scan. But how can you analyze them with speed when it comes to improving the patient's outcome? Manual effort can be very slow. So if you have an automated system, then that improves the speed to which you can diagnose this patient's condition and also be able to move to treatment. Now, when it comes to the current or the fidelity of the simulation. I think, yeah, we still have the issue of validation, which again is something that we always have to deal with when it comes to computational modeling. But I think with experts insight and expert input, both from the computational mechanics point of view and also the clinical point of view, we are able to obtain very good validation. But I think ultimately AI is not going to replace all we do. It's just going to help us to do more and to speed up what we do. So there will be that element of expert validation, expert cross-checking before decisions are made. But of course, it means that we can do more within this a shorter amount of time.
Yeah, thank you. And I have a question from Rudolf Helmuth here, actually 2 questions from him. The first one is, can you objectively demonstrate the cost reduction of mathematical modeling and computational simulation in the R&D of medical devices, especially in the context of regulatory clearance? I think that maybe Alex can answer this question. In previous presentations, even in RAPS for the regulatory folks and in other international fora, we came up with some numbers showing the reduction of costs. Alex, would you like to add something?
Yes, that's an interesting one. So, okay, if you don't ask me for the quotes for where this comes from, because it's probably a lot of areas, but there are. So the numbers that I know is that some people say that you could recover about 5 times the investment you do in modeling and simulation within your R&D cycle. Other figures that I'm aware of is that you can advance up to 2 years your launch of a particular product, so you can get 2 years earlier, and that when you combine it with the sales that you could do during that period is not just what you save, but is the what you would say time to revenues, right? Improvement. Now I will put down on the chat, there is some additional pieces of work that were developed a couple of years ago, and some new work that we are doing at the moment at CEiRSI, where we are doing a much more detailed exploration of those numbers, but also not only looking at the benefits for your particular device as a company, but more broadly in terms of economic impacts to in terms of the benefits associated with jobs, benefits associated with the NHS in the case of the UK, so sort of indirect benefits of incorporation of modeling and simulation, and also in terms of benefits to the reduction as well of risk. So the point I'm trying to make is the element of recovery, of course, is multifaceted. It's not just a single angle to that.
Return of investment is very important for the manufacturers, right?
Yeah, and the other thing is that, and this is a bit of a comment and a plea as well to all of those of you who come from the industrial sector, is there is a view that this information is also quite important in terms of providing a competitive advantage, right? So as you can imagine, many companies hold these figures very tight and close to their chest. On the other hand, I would argue that if we were able somehow, and I recognize it might not be in a simplistic way, but companies would start providing more evidence, those benefits, that will move the dial for the whole sector, and therefore as well will be supporting regulators and regulatory agencies in their decision to move forward. So there's a bit of a balance to be stricken between the sort of confidential element of some of these figures, and also the opportunity to move the dial further and everybody benefiting from it. I hope I answer.
Yes, totally. And something that Mark Horner put on the chat as well, that we find that companies derive more more benefits from early time to markets versus reduction of R&D costs. So this may be related to a particular manufacturer strategy in terms of competition levels as well, right? So very good comment from Mark. And then the next question from Rudolf is how do you compare surrogate models based on deep neural networks, which are black box models, versus more traditional smoothed interpolation functions like Kriging, whose regions of credibility to validation points might be more evident? I think that Adesola can answer this question.
Yeah, thank you, Rudolph, for the question. It's a very interesting one and a very important one. From my point of view, of course, it's still a work in progress. There are lots of research emerging in this area. However, what I want to say is, yes, it is true that when you look at the smoothing interpolation functions, they are very trustworthy in terms of uncertainty. So in terms of AI or using surrogate model, they are very good when it comes to scalability. So essentially, the idea going forward will be an hybrid approach, such that the AI or surrogate model doesn't necessarily replace everything we do in physics-based model, but we are looking at a way where perhaps rather than running 1,000, or let me use 100% of numerical simulation that might take a long time, could you run 50% and then the remaining 50% will be based on AI generated results or surrogate model generated results? That's the whole idea. But of course, as we progress with further research, we get to a point where we have better validation, better trust in what we get from our surrogate model. We can then begin to say, okay, let's scale that for higher, maybe 80 to 20% ratio. But I think the idea here is, if you want, that uncertainty quantification, then we then use the traditional method. When it comes to scalability, we want speed, we want to get things done quicker, then we can use the AI element of that. But the idea is to be able to find a blend, proportionally, not to 100% replace what we're doing with traditional physics-informed method.
Thank you, Adesola. So that was our last question. We are arriving to the last
Marta, Alex
yes.
hand is up.
Yeah, Alex?
Yeah, can I do a very quick plug on something?
Yes.
So,
yes.
So I'm coming back to the question of economic impact, okay?
Yes.
So an area where this community can help us, and all of you who have been here on the meeting, is I just put on the chat a questionnaire that we are running at the moment with one of the best think tanks in health economics that is worldwide, the Office of Health Economics. And they're actually trying to refine the figures that I mentioned before, and particularly the figures in the economic impact document that I just put on the chat. What I need from you and from people that you could engage in the same help is to answer as many times as possible that question. And we are not going to be asking you things about finances in there, so you don't need to worry. This is mainly for practitioners and people who are actually working in doing sort of guesstimates about adoption across different areas. If we have that with their economic knowledge and some of the econometric modeling that they're doing, it's possible to estimate some of the figures that you're also asking us to provide back. And I think that unless we do it with people who know about the econometric modeling like they do, and also with our understanding on the different aspects which are more of the domain, and in an anonymized manner, that's probably the best shot we have at starting to produce figures. The report will be publicly available, so everybody will benefit of that, like the other 2,000 people who download the other reports so far. Thank you.
Thank you, Alex. Well, with this, we are about to conclude our session. I would like to thank to all the panelists today and the rest of the team that have been contributed a lot in all the discussions that have been contributed, even with the paper that will be submitted for publication before the season's holidays. So stay tuned. We will be keeping you all informed about this paper, the upcoming paper. And thank you for extraordinary participation of all the members of the Pilot 4 members. And I thank the companies that supported as well, supported the pilot, our sponsors, the UK CEiRSI, the Innovate UK, and MHRA that have been a fantastic partner in this pilot project. Thank you everyone for your time today, and we'll see you in the next pilot presentation.